Welcome. Folks are starting to file in here. Good morning. Thanks everyone for joining us. All right, so yeah, my name is Dylan Stern with the Delta Science Program, Delta Stewardship Council. You are here today at the 7th and final seminar this year for the Delta Research Award Seminar Series. And we have a really great one for you today. The theme is multi-benefit wetland habitat and Delta food webs. And so I hope you're all ready to nerd out on two fascinating environmental DNA monitoring projects. Next slide, please. So please add your name and affiliation to the chat when you get a second. We have two polls, one at the beginning, one at the end. Please take a second to fill that out. And we really just use that to continuously improve our events like these. So thank you so much for that. And just so everyone knows, the seminar is going to be recorded and posted to the council's YouTube channel in case you want to share this with anyone you know who was unable to attend. And we really want to encourage participation and discussion, so please write in the chat box. And if you have questions, add them to the Q&A box specifically, or you can raise your hand and I can promote you to speak if you'd rather do that. The Q&A box, you won't be able to see the questions appear until we decide to answer them live. So just don't worry about that. Okay, so let's get started, everyone. Yeah, next slide, please. Oh, no, go back one, sorry. So first I'd like Megan to introduce herself quickly. She's been instrumental in supporting these seminar series. So please give us a quick introduction, Megan.
Yeah, of course. My name is Megan. I'm the state fellow working with Dylan in the research funding unit. I'm super excited to have been on board in creating this seminar series. So I hope you guys enjoy.
Megan. Okay, so the seminar is open to the public and it's recorded. The format is that presenters will have 30 minutes to both present and then immediately answer questions. And we should have some time at the end for discussion as well. We want to encourage that participation and discussion. So add your questions and answers to the Q&A box, not your answers. So there are no bad questions. Just want to be clear about that. So be brave and ask away. And now I'm going to go ahead and hand it over to our Deputy Executive Officer for Science, Henry DeBay.
Thank you, Dylan. Thank you, Megan. And thanks to our public participation team who's behind the scenes making this whole webinar happen. Good morning, everybody. As Dylan mentioned, I'm Henry DeBay. I'm the Deputy Executive Officer for Science at the Delta Stewardship Council. And I'm also thrilled to welcome you all to today's seminar, which is going to be highlighting two projects funded by the Council as part of the 2021 Delta Research Awards. And as Dylan already said, but if you're just joining us and haven't already, please introduce yourself and share your name and affiliation in the chat. So first, I did want to provide a bit of background about the Council and the Delta Science Program. So the Delta Stewardship Council, including the Delta Science Program, was created by the California Legislature through the 2009 Delta Reform Act. And the Delta Science Program was charged with providing best possible scientific information to inform water and environmental decision-making in the Delta. And one of the ways that we provide that best possible science is by funding scientific research in the Delta, including through competitive research calls like the 2021 Delta Research Awards. And it's worth highlighting that our science funding efforts work hand in hand with two key tools pictured here on the slide, the Science Action Agenda and the Delta Science Tracker. So the Science Action Agenda, if you don't know, is a collaboratively developed document that identifies management relevant research priorities. And the Delta Science Tracker is a web-based tool that inventories research projects across the Delta to promote communication and collaboration. And of course, the purpose of today is to go beyond simply funding science, but ensuring that the science, that this science, including the results and the management implications, is communicated broadly. So next slide, please. Okay, so I promise we're getting to what y'all are here for momentarily, but lastly, I do want to just share a couple of announcements that you may be interested in or know folks who are. So first, Dr. Kenny Rose, a member of the Delta Independent Science Board, will be making a presentation this Friday summarizing a recent DSP-facilitated review of the U.S. Bureau of Reclamation's Fish and Aquatic Effects Analysis for the long-term operations of the Central Valley and State Water projects. It's a bit of a mouthful, so tune in if you'd like some more information. But yeah, Dr. Kenny Rose was the lead author for that review, so it should be interesting. Second, registration for the 2024 Bay Delta Science Conference is now open. The theme of this year's conference is Cultivating Connections for a Dynamically Changing Environment. And the event will be taking place September 30th to October 2nd at the Safe Credit Union in Sacramento. So please consider joining us for these events and or spreading the word. Next slide. So as Dylan mentioned, this is the 7th seminar of the series and the last one scheduled for now. Each seminar that we've been hosting features 2 presentations on a shared theme from the 2021 Delta Research Awards. And today's seminar theme is multi-benefit wetland habitat and Delta food webs. And each project's key research questions, findings, and management relevance will be summarized in information sheets and available on our website. And recordings of all of these seminars are also available on our YouTube channel. So while this is the last seminar for now, in the spring of 2025, we will pick up the series again and highlight 3 remaining 2021 Delta Research Award projects. One by Dr. Renee Henry and team, focused or titled Reorienting to Recovery, a project that's focused on life cycle modeling and for the purposes of salmon recovery. We'll also highlight Dr. Russ Perry from the U.S. Geological Survey and colleagues and their project on developing a spring run Chinook salmon juvenile production estimate. And last but not least, Dr. Rebecca Buchanan from the University of Washington and their team on standard operating procedures for predator detections from telemetry data. Next slide. So again, today's seminar theme is multi-benefit wetland habitat and Delta food webs. And as many of you know, wetlands are iconic and multifaceted ecosystems. And so to assess and manage these very special environments in the Delta, scientists are exploring them from every angle, from understanding the challenges that they face from sea level rise to the effects that restoration is having on wetlands, to advancing new non-invasive monitoring tools like environmental DNA or eDNA, which you'll hear about more from today's speakers. So today's first presentation is by Dr. Michelle Jungbluth of San Francisco State University, and Dr. Jungbluth's team characterized species diversity of restored wetlands in the upper San Francisco estuary and delta at multiple levels in the food web and in the water column from bacteria to fishes. So this research addressed science action, I should say 2017 to 2021 science action agenda, science actions 3, 4, and 5. So with that, I'd like to hand it over to Dr. Jungbluth to take it away. Thank you.
There we go. I think there's a small lag. Can everyone hear me? Lovely.
Yes.
All right. Thank you for that introduction, Henry. And thank you to everyone for joining us today for our Delta Research Award seminar. Today, I'm excited to share with you our project for Microbes to Zooplankton, what defines a beneficial wetland, and I'll be presenting with my graduate students, Eric and Anthony, but also would like to mention my co-PIs, Dr. Jason Hassick from ICF and Dr. Wim Kammerer, who assisted with developing and carrying out the project. Before I get started, I also want to thank our funders, of course, at the Delta Stewardship Council and the U.S. Bureau of Reclamation. This project, they probably didn't know, was my first project to be funded as the lead PI, so I really appreciate getting the opportunity to do this exciting work and have it be my first big project. I'd also like to thank all the folks who contributed to the development of the project throughout the process. We regularly connected with stakeholders and collaborators to pick research sites, design the study, make sure we did our best to maximize the information gained from all the efforts that had to go into collecting the samples and working through the analyses. So to give you a little bit of background about our project and why it's relevant, our work is highly relevant from the perspective of the wetlands that provide essential fish habitat across the San Francisco estuary. Currently, the wetlands we have today represent what is left after losing about 80% of historic wetlands across the San Francisco Bay and Delta. The maps here show sort of historic wetlands in green and modern wetlands as of 1998, we'll say. Might have been the nicest plot we could find at the time, but showing just the different colors illustrate modifications to those wetlands, things that are diked, things that are agricultural or developed islands, that sort of thing. So clearly a lot of changes have happened compared to historic wetlands. And wetlands are considered to be essential beneficial habitat for fishes for a variety of reasons. They provide things like shelter, enhanced invertebrates and plankton food resources. Those are just some examples. We know that in the water column, we call them pelagic organisms in the estuary, include various native and non-native fishes have experienced sharp declines in their abundance. And one reason that they have declined is because of declining food web resources in the water, which affect fish abundance. When we look at wetlands and when wetlands are being restored, they're often monitored, but that monitoring happens mostly above the water surface. Although in our Bay Delta estuary, there's a lot of fish monitoring that also happens. But traditionally, folks look at things like benthic macrofauna, benthic invertebrates, vegetation, and adult fish in the water column, but they rarely look at things like bacteria, plankton, or the larval fishes that require this habitat for essential nursery habitat and their food resources. So the research aims, the aims of our research have been to sequence the diversity in the microbes to zooplankton living and restoring wetlands across the San Francisco estuary, mainly the northern estuary. You'll see our sites in a few minutes. To identify the biological indicators of the status of restoration, mainly comparing young versus mature wetlands, and of the benefits being provided to higher trophic levels, primarily the fish. And the driving questions of our research have been, do wetlands in different stages of restoration provide different food resources to the fishes that utilize them? And is there a succession in the biological communities over time that could be used to indicate the progress of restoration that can be monitored by simply taking a water sample? That's sort of the pie in the sky ideal situation. Can we take a water sample and say, this wetland is doing what we want it to do? So as a visual of our overall approach, we're using DNA sequencing to characterize the biological diversity in early, intermediate, and mature stage wetlands. This lovely figure was developed by my graduate student, Anthony, who you'll hear from in a moment. And it shows sort of the different levels of biological diversity and how they're connected via the food web. And so in our project, we're doing community DNA sequencing of the bacteria, the bubble A, the phytoplankton and microzooplankton, bubble B, which are both prey of the zooplankton, and bubble C, the zooplankton are prey of the larval fishes, of course. To do the community DNA sequencing of those first 3 levels, we collect water samples and put that onto a filter, and then get the DNA off that filter to characterize the species that live there. And then we also collect net samples for the larger zooplankton organisms and sequence the diversity in the zooplankton fish prey. And then we're also comparing that to what's in the diets of the fishes, doing dietary DNA sequencing of larval fishes collected with plankton nets. To bring it back to those science action areas that Henry mentioned, we're mainly connected to science action area 3, 4, and 5. That is developing tools and methods to support and evaluate habitat restoration, evaluating the performance of restored areas on a landscape scale. Science action area 4 was improving understanding of interactions between stressors, managed species, and their communities, and determining how restoration actions will affect native fishes to adaptively guide management decisions and restoration design. And science action area 5 was modernizing monitoring. So at the end, I'll sort of bring it back to these just to sort of bring it all together. Now I'll turn it over to Anthony to talk about his work on the food web component of the study.
Hello, good morning. So our collection sites were in the San Pablo Bay and Suisun Bay in the upper San Francisco estuary. As you can see on the map, the colors represent the different stages of restoration of the wetland collection sites. So our collection was done between February and April of 2022 and 2023. We used 150 micron mesh net for the zooplankton toes behind the boat, and 500 micron net for the larval fishes. Now this was done in the wetland channels during the ebb tide when the water is coming out of the wetlands. And we immediately process and preserve the samples in 95% ethyl alcohol. Next slide. So the dissections of our 3 species of focus for longfin smelt, Pacific herring, and prickly sculpin. The dissections of the digestive tract, they're done under microscopy with tweezers and insect pins. The total length of the larval fish is typically between 3 to 12 millimeters, which is roughly equivalent to an eyelash or 2. I'm also processing the zooplankton by taking subsamples and concentrating them onto a small filter. I'm currently in the process of extracting the DNA of the larval fish and zooplankton and building the PCR library before we sequence them in the future. Next slide, please. So our preliminary results for the total larval fish abundances for all species collected. As you can see, 2022 was a dry year. 2023 was a wet year. Our collection was mostly dominated by yellowfin goby, Pacific herring, and prickly sculpin. In 2023, most noticeably an increase in abundance of Pacific herring. You can see in 2023, you also definitely an increase in prickly sculpin and longfin smelt as well. Next slide, please. So our preliminary results for the native and threatened longfin smelt. So 2022, there was less abundance, and there were a few of the wetland sites where we did not collect any. In 2023, the wet year, you can see that there was an increase in abundance, and we did collect longfin smelt at every wetland collection site. So it definitely spread into the Petaluma region of the San Pablo Bay. Thank you. I'll let Eric continue with our presentation.
Thank you, Anthony. So my component of the project is to identify which microorganisms are present in these wetlands. So I sampled at the same locations and dates of the larval fish samples, except for I collected my samples with water samples using sterile bottles, which was then put on ice to be taken back in the lab for processing. Once taken back to the lab, you can go to the next slide. Once taken back to the lab, we used a method known as metabarcoding, which is a large-scale taxonomic identification of complex environmental samples via analysis of DNA sequences for short regions of one or a few genes called DNA barcodes. I attempted to simplify this metabarcoding process into 4 steps, starting off with DNA extractions. So after we've gone out and collected our samples, we have a lot of material within our water samples, and we want to isolate the DNA so that we're only looking at the DNA. So we do that via DNA extraction. Step 2 involves amplifying the DNA with PCR to target different organisms. Step 3, we then sequence with high throughput DNA sequencing, and which leads to step 4, which we then classify the DNA sequences to get taxonomic identification. Go to the next slide. And the main point of all of this, again, is to see if we can find indicators of wetland conditions. And there is literature on this demonstrating which conditions promote specific organisms. But I, of course, am interested in the microbes. So an example of some key microbes include like algae or phytoplankton or other photosynthetic organisms that might tell us something about the light conditions. Or maybe we see denitrifiers, which tell us something about the dissolved and particulate organic matter within our water column. So go ahead and go to slide 3. And so far, we do have sequencing results from the 2022 water samples. This is a NMDS plot, and the main thing you need to understand here is each point represents a sample that was taken. Each point is color-coded by wetland and shaped by region. The first thing that we notice is that there is a salinity gradient from high to low. So the samples that are more west, or the wetlands that are more west, have a higher salinity versus the samples that are taken more east, which have a lower salinity. I am going to show you certain regions with the red box here. Starting off with the Petaluma region, we do begin to see groupings, which means that within this Petaluma region, these samples all contain similar organisms. We do see the same thing with the Napa regions, and same thing with the North Sassoon regions, a little bit more stretched out. With the East Sassoon regions, a little bit more stretched out. But here in the middle, we do see more closely related samples between Thule Red and Fire Island. Even though we have connected them or put them into 2 different regions, they do share similar community based off of their location or their geographic location, similar location to one another. Next, I'm going to show you guys a RRA graph, or a relative read abundance graph, which goes into a little bit more detail of what specific organisms are within our sample. I have only shown you here the Napa region and the Southeast Sassoon region highlighted in blue and yellow on the map. The first thing I want to point out is within the Napa region, we do see a lot of this purple organism kind of dominate, the alpha proteobacteriae. Second thing I want to point out is within the Southeast Sassoon region, looking specifically at Winter Island and Browns Island. So even though these 2 wetlands are very close to each other in proximity, we do start to see a little bit of difference in terms of what term it seems to dominate within our sample. One thing that we notice is within the Winter Island, we see a lot of the yellow, or the bacteroidea, compared to the Browns Island. Another thing that we notice in like this teal bluish color, the Brown Islands seem to have a lot more of the verruco microbiae. So these are just some things to start to look at, and we are just, these are just first looks at to see how these sites differ. We will be conducting a little bit more in-depth analysis to connect these indicator species to wetland conditions. We also have samples from 2023 that we want to look at and compare to the 22 samples, which is looking at a wet region or wet season versus a dry season. So there's still a lot more to do, but we're well, I'll just end it there. Thank you.
So in addition to all the core sequencing data that is the primary sort of data output of the project, we have all sorts of associated environmental data to relate with the biological assemblages, including some nutrients that we collected in 2023 to get an idea of what nutrient levels were across the sites and the bottom-up support for higher levels of the food web. We also have water quality conditions collected with the sonde, like salinity, turbidity, temperature, chlorophyll, and oxygen levels. And we also have counts and visual ID of the zooplankton assemblages, and to look at how not only the assemblages differ, but the abundances of the different prey items available to fish and how that changed over time and space. So to wrap it up, the next steps are to finish sequencing, which Anthony and Eric are working hard to do in the next few months, and sequencing the larval fish diets and zooplankton samples and connect those diets to the wetland stages and the occurrence of indicator species in those wetlands. Sequencing the 2023 water samples, get the bacteria, phytoplankton, mycozooplankton indicators of wetland conditions, and connect all of that biological diversity with conditions and assess for indicators. To bring it back to the management implications in the science action area, so relevant to the action area 3 and 5, the water sampling and sequencing are a non-invasive water quality monitoring technique. So ideally, what we will get is a list of biological indicator species that could be used as tools to support monitoring and restoration efforts in the future. In addition, surveying the DNA in each wetland will provide a biological baseline information about what is there across all of these wetlands. And the sort of outcome of classifying the DNA is representative of what's in the database at the time, but as that database grows, you can reclassify the DNA and get more information in the future. So it's invaluable to have these data sets for so many different, unforeseen things in the future. So I think having this is really exciting that we can use it in the future. Relevant to Science Action Area 4, we aim to improve understanding of the interactions between managed species, mainly our native fishes, and their communities of prey and other biological organisms, and with an aim to determine how restoration actions will affect native fishes and help adaptively guide management decisions and restoration design. So by identifying indicator species in these wetlands, ideally, if we see that certain wetlands sites or regions are performing better through their indicator species, ideally in the future, we can take a water sample and look for those indicator species and be able to get, put that together with other pieces of information to guide where to prioritize future wetland restoration or restoration improvements. So hopefully that all sort of makes sense. And with that, I'd like to thank you all for your attention. I'll also highlight some additional supports or sources of student support that are listed here. And hopefully you have some time for questions and discussion. Thank you.
Absolutely. Thank you very much, Michelle and the rest of the team for that really great presentation. Very cool data, and appreciate the specific call outs for the science action agenda and the management implications of this work. So let's see, I'm just going to look in the chat. We do have a question. So Andrea Schreier is writing, it looked like Ryer Island had a more similar bacterial community composition to the Napa area wetlands than nearby wetlands it was grouped with. Any ideas about why that might be? Also, great talk.
Thanks, Andrea. Appreciate that question. Yeah, I think that's where the more in-depth connection to the environmental data, is it just salinity driving these patterns, or is it, things like the nutrients, or is temperature an important factor? This is where those sort of deeper analyses are going to come into play and help us understand not just what's different, but what's driving those differences. So, you know, in terms of if you picture sort of the water flow, I guess, maybe Ryers, you know, that's probably the most adjacent to the Napa area, but it's still pretty, you know, geographically distant. So, yeah, I can't really say exactly why at the moment, other than we'll see. We'll see what's driving those differences. So far, salinity seems to be the primary driver of differences across the broader scale, but I think we'll be able to see more specific things as we do more in-depth analysis.
Thank you. Not seeing any other questions right now, but if you do have any, please add them. I will ask, and sorry if you covered this, but I think in some of the first slides, you highlighted the restoration sites that you used and also the level of restoration for those sites. I forget the criteria, but how did you determine that level of restoration?
That was tricky and it is a little bit wobbly, I'll admit. But so that was where conversations with folks at NER, the National Estuarine Research Reserve, Stuart Siegel helped a lot in sort of talking me through, especially like the Petaluma and Napa sites he was very familiar with, trying to, you know, if we had to put them in a box, what generally are they most similar to? But yeah, I'd say generally recently breached sites that are mostly sort of open mudflat types, like Cullen and Ranch was very representative, like open water, you know, not a lot of channelization, not a lot of vegetation. Those got classified as early stage. In contrast to mature, being highly channelized, vegetated, been there for a long time, generally. Some of them, so in the Suisun region, there's not a lot of restored sites. So a lot of those are just mature reference sites more than restored site or like technically restored. We didn't have a lot to work with there. And we also didn't have a lot of intermediate stage in the Suisun region. It was either early or mature and not much in between. So so it was what we, you know, lots of conversation with folks were sort of led us to the, well, at least we can do the early and mature and maybe have a couple examples of the sort of intermediate and see if we can pick out a gradient. Yeah.
Very cool. Thank you.
Mm-hmm.
So we got a couple more questions in the Q&A. So Tamar Kraus from USGS is asking, can you gain insight into contributions on a biomass or carbon basis for bacteria / phytoplankton / invert / aquatic veg from this data set?
Let's see, biomass. Let's see. I would say with the bacterial sequencing, there's been a lot, you know, bacterial sequencing has gone on for much longer than the larger multicellular organisms. And generally that roughly translates to biomass very roughly. So the relative, you know, these things are relatively more abundant, relatively more biomass in the groups that are more abundant. And that holds generally well for larger species with some caveats that there are, with any DNA sequencing, there can be primer biases. So we always have to keep that in the back of our mind. Are we missing anything that we know is there with the primer set that we chose? We do vetting beforehand to make sure we use the most broad primer set we can for the species that we're trying to target, or the groups of species that we're trying to target, but they all have their sort of limitations. I guess as far as, oh, here, I'll pull it up to make sure. Contribution biomass, carbon bases, phytoplankton. Phytoplankton are tricky. I mean, just generally the the relative proportions of things do translate, especially for the more abundant things. Once you get into the less common organisms, it can be hit or miss. So with, you know, the top 50 or the top 25, they're pretty reliable as far as if they're shown to be abundant, we know that they're relatively pretty abundant in the environment. Or yeah, hopefully that answers your question.
Yeah, thank you very much. So we got 3 additional questions here in the Q&A. So an anonymous attendee is asking, is the fish diet analysis being done with only microscopy, or is DNA sequencing involved in that portion of the work as well?
Sorry if that was unclear. They're dissected under the microscope for DNA analysis. So we aren't doing microscopic diet analysis at all. They're just because they're, you know, the size of an eyelash or a couple eyelashes. They're very small fish. They get dissected very carefully under the microscope with sterile tools, and then the guts get extracted and sequenced.
Yeah, seeing that slide. Sorry, go ahead.
Yeah, it's quick additional comment. I mean, visually under microscopy, I do take notes on the color of the digestive tract, whether it's transparent, light brown, or dark brown. There's also some digestive tracts where visually seems that there are copepod eggs that I make notes of, or other dark masses or dark specks that after we sequence, we'll look at that more closely.
Yeah, and when you're doing the dissections, I know you try not to tear them open because we don't want to lose DNA before we get it in the tube. So it's kind of hard to identify what's there without opening guts visually. So he has to sort of do his best to just give it a general looks full, not full sort of classification.
And kudos to all of that microscope work. It was giving me flashbacks to picking out forams from sediment cores. So yeah. Okay, we have another question from Cheryl Patel. Good to see you, Cheryl. Haven't talked to you in a while. Do you feel like you're able to get a good picture of the organism diversity in your community samples based on the currently available DNA sequences in databases? Also, this was a great talk.
Yeah, thanks, Cheryl. Nice to see you here. Yes, and I think so far with the sequences that we have, it's the bacterial assemblage, which was 16S gene sequencing, which some may be aware is much more vetted than some of the other markers that we have to work with for eukaryotes, the larger organisms, multicellular things. So I think for what we have so far, yeah, I think we have a good picture of the microbial diversity with that data set so far. So we'll see once we get more sequence data how that all looks. Thanks, Cheryl.
Yeah, thanks. And then we have a question from Elijah Portugal at CDFW. Sorry if you covered this and I missed it, but what were the primary factors considered for classifying the age or condition of the wetlands in the study?
Covered this in the beginning of the Q&A, but we can I can touch on it again. It was difficult, and it is a little bit wobbly because that, as you may know, two wetlands next to each other may progress differently if they're breached at the same time. But in general, the main thing we're going to be able to compare is the early stage versus mature wetlands. And the early stage was mainly breached somewhat recently, last 5 years, I think most of them. But what they are out in the environment, they're mostly open mudflat type with very little vegetation, very little channelization. And that's sort of the main thing, is the lack of vegetation, channelization, and sort of open. So good examples are Cullinan Ranch is very open, not a lot of vegetation, and Wings landing was very recently. I think it was breached like the year before we started sampling. And that one's very open. And then the mature ones, we only had a few mature restored sites, mainly in like Napa and Petaluma. There were mature sort of reference sites, mainly in the Suisun region. So it's going to be hard to compare like restored mature versus, well, no, we'll be able to compare restored mature in different regions to reference mature in other regions. That was where like we met with folks Fish and Wildlife and at the NER, National Estuarine Research Reserve, to sort of figure out, okay, what can we do? What are the best sites to compare? What do we have to work with if we want mature sites in these different regions? And yeah, got to work with what we got. But I think it's still really exciting to have all this data. Yeah.
Thank you. I'm not seeing any other questions, but I do have one, so I'm going to take this chance to ask it. And forgive the ignorance here, and this is going to make some DNA experts on this call probably cringe, but including you all. So in terms of going back to just the extraction of the long fin smelt, like gut content analysis, why can't you just take that whole thing, take the whole sample and sequence it? I mean, is taking the contents of the gut out to create efficiencies down the road in terms of like separating out the DNA? But I mean, why not just, I hate to be so crass, but blend the whole thing and then sequence it? And maybe that just shows my lack of understanding of the process.
Yeah, that's okay. Sorry, we probably weren't clear there. We pretty much do. Like, so he removes the whole gut tract. He doesn't open it up or anything. He takes the whole gut tract off the fish and puts it in a tube, and then that gets extracted and sequenced. An alternative would be to just throw a whole fish in the tube and extract and sequence that. Part of the challenge with that is you end up getting a lot of fish DNA. And when you're working with eukaryotes and trying to sequence other eukaryotes from their diets, you have to use certain gene regions that will also amplify the fish DNA. So if we can minimize how much fish DNA is in the sample that we're trying to sequence, that gets us a bigger signal of the actual prey that we're looking for. So that's why he has to dissect them under the microscope to remove the body of the fish and everything from that sequencing signal. We also work with blocking primers that reduce the signal of the fish as well. It doesn't totally get rid of it, but it does reduce the amount that we end up sequencing. So that helps too, but yeah.
Great. Thank you so much. Appreciate that explanation. Okay, well, we should move on to our next presentation, but thank you again, Michelle, and to the team for this great presentation and Q&A.
Thank you.
Okay, next slide, please. So our second presentation today will be by Dr. Ravi Nagarajan at UC Davis, as well as Leigh Sanders. And their project, which you'll hear more about shortly, use single species and multi-species environmental DNA approaches to monitor fishes and the broader food web at tidal wetland restoration sites and paired reference sites. And this research addressed Science Action Agenda, Science Actions 3 and 5. And with that, I will hand it over to you, Ravi.
Thank you very much. Can everyone hear me okay?
Yes.
Okay, great. So thanks very much for the opportunity to present today. And we're presenting our preliminary analyses from this project. We still have some data analyses ongoing. So this is a kind of partial picture of what we've seen so far. And next slide, please. So I'll start by acknowledging our funding and our collaborators. So I would like to acknowledge the Bureau and the Delta Stewardship Council for funding. And for our collaborators, we have many different collaborators, so we need to acknowledge, but I'd like to especially acknowledge Stacy Sherman, who leads the fish restoration program, who's just like been a key essential collaborator through the process. And I also want to shout out to Michelle. Really enjoyed your presentation, Michelle, and her team, including Eric. We collaborated with them when we were in the process of planning these projects, and throughout the projects, they helped us with sample collection. And it's just been a really great collaboration. So thank you. And next slide, please. So Michelle had a very similar slide, so I won't go into detail, but I mean, the take-home message here is that there's been this huge, massive decrease in tidal wetlands, and one way to mitigate that loss is to restore some of them. And so there's been many thousands of acres restored over the last several decades. But that necessitates the need to monitor what's happening there and to monitor regularly over time. And so our project is a monitoring focused project. And we're especially interested in using environmental DNA or eDNA as a tool for monitoring, and basically to provide a new tool, an efficient tool that's non-invasive to managers to give them more information, more comprehensive information about tidal wetlands that can also be applied in many other contexts as well. So we're especially interested in monitoring fish species, so entire fish assemblages, but we're of course especially interested in knowing how well and if we can detect listed fish species like the delta smelt, longfin smelt, and winter and spring run Chinook salmon. So that's one of the benchmarks that we're looking for, is like, are we going to be able to detect these things with our methods? Yes, so our project is using these eDNA methods at tidal wetland restoration sites in a monitoring context. So next slide, please. And the basic aims for our project were to monitor fishes at paired restoration and reference sites using an eDNA approach, a non-invasive approach. And Leigh, our graduate student, will talk a little bit more about the details of that later. On the right, here's a picture of one of the restoration sites. I think Leigh possibly took this picture. We did this sampling in collaboration with existing monitoring. So that was an important detail for us, is that we're not just doing this kind of like on our own, but we're trying to benchmark our data to really, solid foundational gear-based monitoring data. We wanted to look across different time points and see if we can see seasonal differences, like see different fish assemblages at different seasonal time points. And also we're trying to be as taxonomically broad as possible while still being really streamlined and efficient in our sampling approach. So I think in Michelle's presentation, she talked a lot about different sampling approaches that really effectively capture different parts of the food web. And we'd like to do that as well, but we're basically trying to do that through simple water grab samples and just looking at multiple different DNA barcodes, which may not be as comprehensive, but could be a streamlined way that you could get a sense of not just the fish species that are present, but also look at the food web, be able to detect dietary items, prey items, plankton, things like that. Next slide, please. So our project is responsive to two main science action agenda areas, similar to Michelle's project. Action area 3, to develop tools to support and evaluate habitat restoration. So we're trying to understand if eDNA can be a non-invasive, cost-effective tool for evaluating habitat restoration, and can it complement existing methods? And also action area 5, to modernize and basically bring new tools to monitoring. And we consider eDNA an innovative technology, and also it can be a really cost-effective technology depending on how you implement it. So we think it has a lot of potential here. And next slide, please. So all of our eDNA sampling has been performed basically in parallel with existing monitoring. So I mentioned that monitoring is so essential to understanding how restoration is going. So we've been collaborating really closely with Stacy Sherman and the Fish Restoration Program. So they monitor multiple different restoration, as well as paired reference sites. So this is in the, I guess, more the central delta near the confluence. And these are the sites that we've been looking at. And Leigh will talk more about the specifics of the sites in a later slide. But we're using also sampling at this paired restoration and reference site approach. And I just wanted to highlight that, these monitoring programs, the existing monitoring programs, are, provide this foundational data, but they have limitations related to incidental take. Of course, there's limitations in resources and time, and just, there's only so much people can do. So eDNA might be able to fill in some of these gaps in some ways. We've also collaborated with the UC Davis Sassoon Marsh Fish Study, and we've done a lot of collaboration with them, but specifically here, we wanted to be able to access and sample at the Montezuma Wetlands Restoration Site and be able to do that in parallel with gear-based monitoring. So just to kind of summarize this, monitoring is essential for evaluating restoration, but of course, resources are limited and there are some data gaps that remain. So how can we fill in maybe some of those gaps? And at this point, I will transition to Leigh to talk through some of our methods and data.
Yeah, thank you. So previously we mentioned that we'll be using eDNA to monitor fishes at these tidal wetland sites in the San Francisco estuary. And some of what I say will be a little bit repetitive because we're talking a lot about eDNA. But I wanted to address again, what exactly is eDNA and how can it be useful for monitoring projects such as ours? So eDNA is DNA that is shed by organisms into their surrounding environment, such as soil or water in our case. So what's really great about this is that instead of directly catching these organisms to ID them, scientists can then instead collect eDNA directly from the environment. So for instance, water samples, making it a really great non-invasive method to ID species. Additionally, this approach tends to be quite cost-effective and sensitive when compared to other monitoring approaches. And we can go to the next slide, please. Okay, so here we have our study sites. And before I get into those, I think it's also important to talk about how our study is defining what restoration and reference sites are. So in 2017, Interagency Ecological Program's Tidal Wetlands Monitoring Team defined restored sites as disturbed wetlands that have been treated through some adaptive management strategy aimed at ecosystem recovery. And they defined reference wetlands as pristine wetlands that can be used as a benchmark for a restored site that is along a shared ecocline and also has similar accessibility to target species. But they also made sure to note that there really is no pristine wetlands in the Delta. Instead, reference sites were chosen if they either had not been diked or drained within the last 150 years, or they had been at least restored to tidal action for over 30 years. So for our study sites, we have 9 in total. 5 of these are restoration sites. We have in the blue, Tule Red, Montezuma Wetlands, Winter Island, Decker Island, and Yolo Flyaway Farms. And we have 4 reference sites, Ryer Island, Browns Island, Web Tracks Island, and Liberty Island. And in the ellipses, like the ovals, you can see how we group together restoration and reference sites. So we were able to collect water samples for each of these sites across 3 different time points between early 2023 and January of this year. And we also were able to pair our samples, as mentioned previously, with gear-based capture surveys from either the Fish Restoration Program or the Sassoon Marsh Fish Survey. Okay, next slide. So after we collected water samples from each site across these multiple time points, we then filtered and extracted eDNA from the water samples. And then we used a metabarcoding approach to sequence our samples to make taxonomic assignments. And we already talked a bit about metabarcoding in the previous presentation, but again, I'll mention it. That basically uses short standardized segments of DNA called barcodes to be able to identify multiple species simultaneously in a sample. For our study, we incorporated 4 different barcodes, specifically the 12S, 16S, CO1, and 18S barcodes. So next slide. Now getting a bit into our preliminary results, currently we've been able to get sequencing results back for 2 of these barcodes, specifically the 16S and the 18S, for 2 out of our 3 field seasons, one being in February and March of 2023, and then August and September of 2023. For the 16S barcode across both of these seasons, we were able to detect 41 species. Of those, 17 were native. And then here in the Venn diagram, it's just showing the number of detections for each season. And so we can see that they shared 33 out of the 41 species detections, so lots of overlap between them. Okay, next slide. Okay, so here was the law, but I'll sort of break it down for everybody. We have a heat map highlighting the abundance of sequencing reads produced for each species within a single sample for the February-March 2023 field season. So starting at the left, we have each of the species detected, and the ones with asterisks to the left are the native species. And all we really need to know about sequencing reads is that their abundance tends to correlate with both biomass and species abundance for eDNA samples. So for this heat map, the darker the red, the more sequencing reads that were produced for that species, and the more abundant that species was likely to have been for that site. Now within each of the larger rectangle, we have each site, and in each rectangle, you'll see there are 3 columns. And those correspond, each column corresponds to a sample. So each site had 3 different samples taken in different locations. And so the first takeaway from this graph that I want to point out is that you may notice that within sites, species detections were not often consistent across those 3 replicates. So it shows that it's really important to take samples at different locations to be able to get a complete picture of what's going on at that site. And additionally, I'm not going to talk about all the species we detected here. I just wanted to sort of show them, but I will point out 3 of our more exciting detections that we were happy to pick up with meta barcoding. First, we saw Chinook salmon at all the sites besides Briar, Thule, and Web Tract Island. We saw longfin smelt everywhere besides Web Tract and Yolo Flyaway Farms. And we were really happy to see some delta smelt at both Montezuma and Briar Island. The Montezuma detection is a very light red. It's a little hard to see, but we did see some there. Next slide. Okay, so now we have a very similar heat map, but this is for the August-September field season. The only thing I'll point out here is that we were not able to pick up delta smelt or longfin smelt with meta barcoding, but we were still able to see some Chinook salmon at both Montezuma, Briar Island, and Thule red. Okay, next slide. So the heat maps were just showing an overview of everything we found. But for here, I wanted to sort of narrow down on the top 10 overall most abundant species for the 16S barcode detected across all of the sites. So here we have relative read abundances that were calculated for each of the species, and we only plotted the values for those top 10 species. So in the top row, we have the August-September field season, and at the bottom row, we have February and March. And for each column, I tried to group together paired reference and restoration sites for easier comparison. So at a glance, we can see that fish species for these top 10 at least tended to vary across seasons and between paired sites. For instance, if we look at flyway for the February-March season, we see lots of Sacramento splittail, which is in orange, but if you go up to August-September, we didn't see any of the splittail in the top 10 there. And then looking between paired sites, especially for this February-March season, we can see some differences. So looking at the brown Montezuma winter pairing, we see lots of splittail in orange and sculpin species in that dark green. But if you move over to Ryer and Thule red, instead we see lots of Pacific herring and definitely not as much sculpins there, which is in the dark green. And then looking at flyway and Liberty, now we see a huge boom in common carp. However, if we start to look within paired sites, you'll see that we're seeing a lot more shared detections in these top 10 most abundant species. However, the differences we're seeing is in the abundance of those species. So if you look at the Brown's Montezuma winter, lots of the same species detected there, just in different levels. So we're going to go to the next slide, but for the sake of time, I'm actually going to skip this slide and the next. They're just ordination plots, basically showing a lot of the same results I've already talked about, just in a different way. And so instead, now we're going to go over to Ravi, who will talk about our 18S barcode. And I saw in the chat about 16S being for fish. Yeah, so some of our 4 different barcodes tend to be specific towards different taxonomic groups, which I should have mentioned earlier. The 16S tends to be more specific towards fish, and the 18S that Ravi will talk about tends to be more specific towards invertebrates.
Yeah, thanks, Leigh. It can be confusing because there's a 16S barcode that's commonly used for bacteria, but we have like a different 16S that we're using for fish as well. So I mentioned in like one of the earlier slides that one of our goals was to kind of see how much taxonomic breadth we could see just with relatively simple eDNA sampling, using the same samples that we used for the fish analyses that Leigh just presented. Can we also see invertebrates, food web organisms, things that would be important for the food web, and things that could be dietary items for listed fish species. So we just have some preliminary data here. This is using the 18S barcode, which is geared towards detecting invertebrates. And this is just data from one season. And what we're looking at is basically kind of cumulative sequencing counts shown on the y-axis. So not broken down by different sites, but on the x-axis showing all the different species that we're detecting. And so what I just wanted to highlight here was the diversity of the organisms that we're detecting, all these different colors, many different eukaryotic, microscopic or small macroscopic organisms. And I just wanted to highlight some of the copepod detections shown here. So those are really important for the food web and as prey items for species like Delta smelt. And we're also detecting some corbicula, the Asian clam. So that's something that kind of indirectly affects the food web. So I think we are starting to, see that we can sort of multi-purpose our water samples. So get more bang for our buck, get more information from a single sample, and detect some food web species. So there's more work to do here. And I think this is probably not fully comprehensive, and we could be missing some things. We could refine our barcodes. We could add in extra barcodes. We could do extra sequencing. We could use other collection methods. But I was really happy to see these results. So next slide, please. So to conclude, like I said at the outset, these are preliminary results. We've got a couple different more barcodes that we're still sequencing. Leigh and I were both in the lab yesterday working hard. We've also got some single species eDNA methods that we're trying to use. And so, for example, we've got some Chinook detections. We don't know which ecotypes, if they're listed or unlisted ecotypes of Chinook salmon. There are molecular tools that we could potentially apply, and we're very curious to try to apply those to the samples, especially if we know there's Chinook. Can we say whether or not it's a winter run or a fall run Chinook salmon that we've detected, or if there's a signal for that? So more to come, but we believe that our results to date demonstrate that we are addressing the science action areas that we attempted to address, that we can use eDNA monitoring as a tool to support and evaluate tidal wetland restoration. And we're really excited about the listed fish species that we've detected already. And we're also just more broadly interested and hopeful about the potential for eDNA monitoring as an additional tool, a modern, cost-effective, really efficient tool for managers. And we believe the future of monitoring is just, it's headed in that way. The eDNA tools are coming online. There's going to be more and more of this stuff happening. We still have some needs and things that need to be worked out. It's certainly not, completely figured out. More understanding and, intentionality about things like data management and the infrastructure needs that we have to be able to store this data, to make it, comprehensible to people, to share it. That's really important. But these things are happening. And on the right, I just highlighted this really recent publication from Liz Bowen at USGS, which used an eDNA approach focused on more longfin smelt in these more marine San Francisco Bay restoration sites. So there's a lot of people working on this, and we're all kind of coming together, and there's this emerging picture of the utility of these methods. So I think there's only one more slide that I wanted to highlight. Thank you. So I just wanted to highlight a little bit of our outreach. So Leigh has actually given several presentations to managers, including at the Tidal Wetlands Restoration Project Work Team and some other venues as well, conferences. She'll be presenting at Bay Delta. But what I wanted to talk about briefly here was our outreach at UC Davis Picnic Day. Every April, UC Davis has this big picnic day thing. If you're not familiar with it, everybody can come on campus, and different groups and labs and entities have booths or displays. So our booth was an eDNA display. So, and Leigh was the one who organized this. And so it was a DNA detectives activity for kids and families, and basically getting them to understand how eDNA metabarcoding works. So you search in this bin and you find barcodes and you match it up to species, endangered species. And we specifically, made a point of providing documentation and we talked about how this tool is being used for this project to understand tidal wetland restoration. So that's some outreach that we're trying to do. And next slide, please. So that's it for our presentation. Thank you very much. We'd love to get your feedback about what we can do to keep our momentum going. If there's certain, really glaring deficiencies or gaps, we'd love to hear what you think about that. I also just wanted to mention, I know there's a lot of interest in involving the Delta community in our research and in our science. I just wanted to let, kind of share and promulgate this idea that eDNA sampling is something that can involve the local community. So there's the citizen science kind of approach where people in the community can go collect samples, and maybe they don't have access to certain sites, but they could provide, it could be a way to integrate the local community into our science. So thank you so much for your attention. We'd be happy to take questions.
Thank you, Ravi, and thank you, Leigh, for that really great presentation. And love the outreach and that picnic, that UC Davis Picnic Day activity. I don't know if that's something you came up with, Leigh, but that's genius. And I think we might need to have you provide a cameo at State Scientist Day. So kudos.
Leigh.
Way to go, Leigh.
Well, I saw Andrea said I came up with it. I kind of came up with it. Someone in the past had done, I found on the internet, something similar, but for older kids. So I redesigned it to be more inclusive.
Well, it's really great. And I think that's the kind of meta barcoding that I think I can do too. Okay, so there's some questions coming in. I did want to just say thank you for the plain language call out boxes in each slide. I know that's not always easy to do, but I think that's a really good thing to do, especially for those on the call like myself who aren't necessarily experts in this field. And I also wanted to take this chance, sorry, I'm not trying to steal your thunder, but wanted to plug a podcast and article that just posted on Maven's Notebook today that includes an interview with Dr. Andrea Schreie, who is one of the collaborators from this project, and who in this interview highlights this project and other eDNA projects. So I'll add the link to that podcast in, and it's part of a series called Science in Short Podcast, but I'll add that link to the chat. And now I will shift to questions. So we've got a question from Lynn Takada. Hi, Lynn, at CDFW. So thank you for the great talk. Lynn's question is, does the density of detections relate to number of individuals, geographic proximity, or how recently the organism may have been present? If so, could you elaborate?
Leigh, do you want to take that one, or do you want me to?
Sorry, you can take that one.
Okay.
I have a lot of windows I'm sorting through on my screen.
Yeah, thanks for the question, Lynn. And yes, generally it's thought that the density, basically the concentration of eDNA which then, is proportional to the number of sequencing reads you get, is related to organismal abundance in some way. So that kind of can vary by system to system, organism to organism, but in general, there's a correlation. What we really want to do in order to really establish that and to have a better understanding that is to compare our eDNA detections to the gear-based catch, essentially, and the gear-based data from the fish restoration programs, the Soon-Mars fish study, and compare that quantitative data to the quantitative sequencing read data we're getting and see what kind of relationships that we have. So I think that's something that we still need to do.
Yeah, and I think especially a lot of studies that I've been coming across, especially for eDNA, metabarcoding, a lot of the sequencing reads that come out tend to be more correlated with species biomass than species abundance. That might also make it a little tricky to actually say how many are there.
Thank you. Okay, we've got a question from Stacy Sherman at CDFW. Thanks so much for the shout out, Ravi. I was wondering if you'd talk a little bit about interpreting eDNA results in the context of evaluating site-specific occurrence of fish in such a strongly tidal environment.
Yeah, hi, Stacey. Thanks for the question. I think it's a great question. It's really important to the field in general, sort of how specific or how spatially and temporally accurate are the eDNA detections. I think that our understanding is, and I'd love to hear other people's thoughts about this, is that in the tidal environment, that really is a complex environment hydrologically. A lot of these places can be relatively well mixed, and that the detections could result from water that is, maybe several hours old that flowed in from an adjacent site. I think one of the ways we can address this is, to really, first of all, we can compare our data to the gear-based data, and if we can look and see if we're just seeing the same suite of organisms everywhere, then yeah, that would suggest that maybe it's not really that specific. But I think in general we don't see that we see some specificity and some differences, different assemblages at different places that make sense with seasons or restoration status or things like that. So obviously more work to do there, but that would be my take on that is we need more understanding of that. And one other thing I wanted to bring in though is that there's been some published work and some interest in the field about basically incorporating hydrodynamic modeling. So modeling that tries to understand, where water is going and, the physical processes that underlie where water goes and how fast it goes, and basically create models that take eDNA data, but relate it to hydrodynamic modeling. So then you can say, we got this detection, but the hydrodynamics tells us that it could have come from this radius of an area. So we'd love to do that kind of work. And we have some proposals that we've written up to try to get that funded. And I think it's really essential, and it'd be really essential, especially in the context of the estuary where we've got this, tidally influenced system.
Thank you so much. We have a question from Julie Leigh, the chair of, and hi, Julie, the chair of the Delta Stewardship Council. Julie is asking, could eDNA be used to detect non-fish animals like nutria in the system?
Absolutely. And Andrea, thank you for highlighting that. And I know that you're very, very interested in this question. And the short answer is yes. And there are existing eDNA approaches that could detect it. Could detect nutria specifically, as well as other invasive species of many different taxa. And we're actively working on some nutria detection methods in collaboration with some other groups. And one of the, I didn't really talk about, but one of the really potentially important advantages of an eDNA-based approach, it can be highly sensitive, and it could potentially detect eDNA from invasive species before they're visually identified or they're seen by other ways. So it could provide an early warning, and you could track an invasion front over time and get a sense of maybe the density of the invasion. So yes, we definitely want to do this.
Great, thank you. Oh, there's another question from Lynn. I was going to ask a question, Lynn, but no, you beat me to it. So Lynn Takata is asking, since white sturgeon is now a candidate for California Endangered Species Act listing, I'm curious if you looked for white sturgeon.
I don't know about other studies in our lab, but I know for ours, it would be great if we could pick it up. But for when we go to collect samples, we're limited to just surface crabs at the very top of the water. So I do know that if there are species who are down below further into the water column, we're not going to be likely to detect them. So far, we currently haven't picked any up with our approach. But Robbie, are we doing anything else in the lab for them or no?
As far as like a single species assay, not that I'm aware of. I do think that we've detected some sturgeon in some of our other eDNA other studies, like other projects. I can't remember the details of that. I remember that one potential complication is that with some of the barcodes, the barcode itself is really similar between white and green sturgeon. So sometimes you have to be a little bit cautious about what you're interpreting there. But I think it is possible to detect sturgeon. It may be that our barcodes, and Michelle hinted at this in her talk, that barcodes can sometimes be, some barcodes that can be biased more towards some species than others. And I think there may be some bias against sturgeon in some of our barcodes. Increasing the number of barcodes or trying different barcodes. We're particularly interested, like in sturgeon detections, might be one approach there. Andrea's mentioning about single species sturgeon detection methods. Yeah, something that we'd like to do for sure.
Right. Okay, we do need to wrap up, but I am going to be cheeky and squeeze my question in. I'm thinking back to the heat map of the species detections earlier in the presentation, and I was wondering, is that something that you've shared with fish experts to just see how that tracks with what they would expect? What they would expect for that time period, February to March, and across that geographic gradient, or not?
Yeah, currently I haven't shared this heat map. Those were made fairly recently, but I have shared other preliminary graphs of species we started to detect with some folks. Specifically, I know there was someone for in the Sassoon Marsh Fish Survey to look at the detections. Overall, they seem to coincide. I know there were some concerns about Mississippi silverside. For instance, they detect a lot of it at their Montezuma site, but they could see that it wasn't one of the more common detections for ours. So they were really curious to learn more about that. But I'm sort of holding off on saying for certain if we didn't detect them or not. It might just be that our 16S barcode wasn't very specific towards them. So when we sequence our other barcodes like the 12S, we'll see how those sort of align with their capture survey as well.
Thanks, that makes sense. Okay, I'm getting indications from the organizing team that we can ask an additional question that has come in, or an additional two. So Tamara Kraus is asking, one, can you let us know where else your teams or others are collecting eDNA data? Is there coordination across research groups? And two, can these data sets provide insight into beneficial versus harmful algae?
Yeah, for the first part, actually, Andrea, it might be useful to have her chat, add something to the chat about this. So we do have multiple projects. We have some completed projects. We had a Prop 1 project where we were collecting samples in collaboration with the EDSM monitoring survey. And we have some eDNA data, metabarcoding data from that as well. And we've got some little projects like testing out methods where we go somewhere and collect a few samples. As far as like a coordination, yeah, if Andrea can speak to this, I think maybe it'd be helpful. I would say that we don't have good coordination, and it's kind of a lot of my impression is there's a lot of like piecemeal. This group's doing this, and sometimes we don't always know what each other are doing. In this case, it was really helpful to know what Michelle and her group were going to do and to try to do things like not be redundant and try to be complementary. And so the more of that we can do, the better. But I don't know if there's like a clearinghouse somewhere that has that. And as far as the HABs, oh yeah, sorry, go ahead.
I was just saying maybe the Delta Science Tracker to some extent could be that clearinghouse, but I agree with everything Robbie said. We were really lucky to make that connection with Michelle and be able to see how our studies could be designed in a way that they would be complementary and to coordinate. But there's a lot of different things going on with different entities. And just to plug a few other eDNA projects going on in different areas that our lab is doing, and I know that there are others. We're developing assays, Sherlock assays for nutria and descended mussels for early warning detection. And I have a student who is using eDNA to examine biodiversity in vernal pools, including at the Don Edwards National Wildlife Refuge, which is in the Bay Delta area. So.
Thanks, Andrea. And about the HABs, my understanding, we are not looking at that directly. I think maybe some of Michelle's sampling is more directly related to this. I don't think we didn't look for it, basically. My understanding is that it can be a little bit tricky to understand beneficial versus harmful versus HABs, I guess. But there are molecular eDNA sequencing kind of approaches that are moving in that direction. And I know I've seen a talk, maybe I'm trying to remember who it was, about that. So I think that's something that we definitely can get from this kind of data. It may need some more refinement to really like accurately discriminate between harmful and beneficial. But I think this is an area that needs investment.
Great, thank you so much. So at this point, really appreciate the discussion, but we should wrap up. Thank you, Ravi, Leigh, Andrea, for that presentation and for that discussion. You're seeing on the screen here more seminars to come in the spring of 2025. So stay tuned for that. And if you're not on our listserv, please consider adding yourself to the council's listserv. Before handing it over to Dylan to truly close this out, I just did want to really thank the team that has been organizing these seminars every week for the last few weeks. So that's Dylan Stern, our program manager for science funding, Megan Nguyen, our state policy fellow in the science funding unit, Emma Askey and Eric Areka in our Office of Public Participation. So a big thanks to all of them for making these things happen and happen so smoothly. And a big thanks to you all for being here. So with that, I will hand it to Dylan.
All right, thank you. Yeah, and just to second what everyone's been saying, thank you to the wonderful speakers today. You all did an amazing job in your science communication. Really appreciate your time and everyone's time and attention. We hope you enjoyed the seminar series. We do have another seminar planned for next spring in 2025, so stay tuned for that. We have all of our seminars recorded and available on the council's YouTube channel, and so check that out. And just thanks again, and I hope you have a great day. Thanks.